Okay, I'm just going to quickly talk you through a couple of different kinds of enzyme reactions. So this is measuring the rate of an enzyme reaction by measuring the volume of foam that is produced. Now this would come up now and again in papers. So here we have a measuring cylinder and we have a set volume of hydrogen peroxide. Hydrogen peroxide is broken down by the enzyme catalase into water and oxygen. When the oxygen is given off, it forms bubbles or it forms foam in this setup. Now, some ordinary tissues, potato and liver, for example, contain the catalase enzyme all through them, so it's on their surface. So if you drop some potato or liver into hydrogen peroxide, you start seeing these foam bubbles. And remember, the bubbles are produced because oxygen is released. Now, in these experiments, you would need to control the volume of catalase that is released into the hydrogen peroxide, and you control it by using a 1 cubic centimetre of, for example, potato tissue. Now, here is something. The foam, whenever it comes up, you can have high bits and low bits, and it's really important that the foam is measured in a consistent way. Usually you measure the maximum height of the foam. So in my example, from here to here would be the maximum height of the foam, whereas from here to here it's a bit lower. But every time you do the experiment, you measure the maximum height of the foam. It's very hard to be accurate because it is just bubbles, and it's very difficult to control, for example, the tissue age, because you don't know how old that potato is. And it's hard to get a 1 cubic centimetre of liver tissue exactly. So some things are very hard to control, but please be aware of this setup. Another unusual little experiment that might be asked is this one. Now, apple cells, so apple cell walls, if you add the enzyme pectinase, the pectinase breaks down the apple cell wall, and then the apple juice is released from the apple cell. So you could have pureed apple and you could add pectinase to it, and you could put that mixture into water baths at lots of different temperatures, or you could add buffers, but you could use this setup to investigate how pH or temperature or substrate concentration affects the rate of reaction. So basically you have your pureed apple and you have your pectinase, and you put them into a filter funnel, and then you collect the juice. So the faster the rate of reaction, the more apple juice will be produced, because more pectinase broke down more cell walls, so more apple juice was released. Now, this unusual practical, you need to consider different variables. So every time you repeat this experiment, the age of the apple should be the same, the type of the apple should be the same, the degree of pureeing the apple should be the same. So you put it in a liquidizer, maybe at such a speed for 2 minutes. How you actually mix the enzyme and the substrate, so the apple and the pectinase, do you stir it 3 times, 4 times? And then this is something to consider. When is the end point? Either of these would be okay, but every time you do the experiment, you need to use the same end point. So you could simply just set this up and record the volume of juice produced after 15 minutes. Or you could set this up and then just record the time when no further juice is produced. I prefer this one, but either method is acceptable. Another variable that you would need to be consider is the time between pureeing and adding the enzyme and then collecting the results. So you puree your apple, you add your pectinase, and you let it sit for 5 minutes before you put it into this setup? Or do you puree your apple, add your pectinase, and add it immediately? Whatever you do, every time you do the experiment, you have to do this in the same way. So I just want to mention a few things then about enzyme immobilization. So again, if you were using immobilized beads, there are things that you would need to control. For example, the age of milk, the temperature of the milk if you were investigating something other than temperature, the bead size. There's lots of different variables in enzyme immobilization. So this is a very popular example here. Milk contains the disaccharide lactose, and it's broken down by lactase into glucose and galactose. So inside the syringe here we have beads, alginate beads, which have lactase embedded in them. So as the milk is poured over the beads, the lactose is broken down by the lactase in those beads into glucose and galactose. So every time the milk is passed through, more and more lactose is broken down into glucose and galactose. In the setup then, you need to have a little bit of gauze at the bottom, and that simply stops the beads from blocking the tip so that the milk can flow nicely and gently into the beaker here below. It's good when you're doing this to slow the flow of milk. You'll actually end up with a faster rate of reaction because it gives the enzymes and substrates more time to collide and form enzyme substrate complexes. So if glucose and galactose have been produced, you can check that out with Clinistics. And remember, Clinistics tests for just glucose and it is pink. And then the more glucose is present, the deeper the colour of purple is produced. So another thing, small beads, if they are in this setup, they will give a faster rate of reaction because they will have a larger surface area, so more of the enzyme lactase will be in contact with the lactose, so you will have more enzyme substrate complexes forming and a faster rate of reaction.